The human immune system is not a single defense mechanism. It is a layered, coordinated biological network involving dozens of cell types, hundreds of signaling molecules, and responses that are precisely calibrated to the nature of each threat. At the center of this network are two lymphocytes, CD4 T cells and CD8 T cells. These two populations coordinate and execute the adaptive immune response, the branch of immunity responsible for targeted, specific, and lasting protection. Understanding them is foundational to understanding infection, cancer, autoimmunity, and some of the most significant advances in modern medicine. This is Case Zero. Blood is composed of four major components. Plasma, the liquid matrix carrying proteins, electrolytes, and nutrients. Erythrocytes, red blood cells responsible for oxygen transport. Thrombocytes, platelets that mediate hemostasis and clot formation. And leukocytes, white blood cells, the cellular foundation of immune defense. It is within the leukocyte population that our story begins. Leukocytes are not a single cell type. They are a diverse family, broadly divided into two functional categories: the innate immune cells and the adaptive immune cells. Innate immune cells respond rapidly and non-specifically to foreign material. They do not distinguish between one bacterium and another. They recognize general patterns of threat and respond immediately. Neutrophils are the most abundant leukocyte in circulation. They are the first responders, migrating to infection sites within minutes, engulfing pathogens through phagocytosis and releasing antimicrobial enzymes. Fast, effective, non-specific. Monocytes circulate in blood and differentiate into macrophages when they enter tissues. Macrophages are longer-lived. They phagocytose pathogens, present antigen fragments to other immune cells, and sustain local inflammatory responses. Eosinophils and basophils handle specific threats, parasitic infections and allergic responses respectively. These innate cells are essential, but they have a fundamental limitation. They have no memory. Every encounter with a pathogen is treated as new. The response is the same whether the body has seen that pathogen before or not. This is where the adaptive immune cells, the lymphocytes, are categorically different. Lymphocytes represent the adaptive immune system. There are three major types. B lymphocytes, produced and matured in the bone marrow, are responsible for antibody production. When activated, they differentiate into plasma cells that secrete immunoglobulins targeting specific antigens. Natural killer cells, despite the name, are actually part of the innate immune system. They eliminate virus-infected and tumor cells without requiring prior sensitization. Fast, but non-specific. And T lymphocytes, T cells, are the adaptive system's primary effectors. What distinguishes T cells from all other leukocytes is antigen specificity. Each T cell carries a unique T cell receptor, a surface protein capable of recognizing one specific antigen fragment. This specificity is generated through genetic recombination during development, creating a repertoire of billions of distinct T cell clones, each matched to a different potential target. This specificity is the adaptive immune system's defining feature. It is also why T cells require a maturation process unlike any other blood cell. All blood cells originate from hematopoietic stem cells in the bone marrow. Most mature there and enter circulation directly. T cell precursors do not. They migrate from the bone marrow through the bloodstream to the thymus, a bilobed lymphoid organ situated in the anterior mediastinum. Inside the thymus, precursor cells, called thymocytes, undergo a development process governed by two sequential selection checkpoints. The first is positive selection, occurring in the thymic cortex. Thymocytes that can successfully engage with self-MHC molecules, the surface proteins your cells use to display their internal peptide content, receive survival signals. Those that cannot are eliminated through apoptosis. A T cell that cannot read MHC is functionally useless. The second is negative selection in the thymic medulla. Thymocytes that respond too strongly to self-peptides presented on self-MHC are eliminated. These cells carry the risk of attacking the body's own tissue, autoimmunity. The result of this dual selection is severe. Approximately 95 to 98% of thymocytes die in the thymus. The survivors, those that can read MHC without attacking self, graduate into peripheral circulation as naive T cells. And at this point, each T cell commits to one of two identities. The commitment to CD4 or CD8 is determined by which class of MHC molecule a thymocyte recognizes during positive selection. MHC molecules come in two classes with fundamentally different distributions. MHC class I molecules are expressed on virtually every nucleated cell in the body: muscle cells, epithelial cells, neurons, hepatocytes, any cell that has an internal protein environment to display. MHC class II molecules are expressed only on professional antigen-presenting cells, dendritic cells, macrophages, and B cells. Phynocytes that recognize MHC class I commit to the CD8 lineage. Thymocytes that recognize MHC class II commit to the CD4 lineage. This difference in MHC recognition directly shapes function. CD8 cells will surveil the internal state of virtually every cell in the body. CD4 cells will interact exclusively with the immune system's own antigen-presenting cells. Two populations, two surveillance domains, permanently fixed. CD4 T cells are classified as helper T cells, though this designation understates their role considerably. CD4 cells do not kill directly. What they do is coordinate the entire adaptive immune response. The sequence begins with antigen presentation. When a dendritic cell, a professional antigen-presenting cell, captures and processes pathogen-derived material, it displays peptide fragments on MHC class II molecules at its surface. It then migrates to a lymph node. There, naive CD4 T cells with matching T cell receptors encounter this MHC peptide complex. Recognition triggers activation. Activated CD4 T cells proliferate, generating large clonal populations, and begin secreting cytokines. These signaling proteins are the mechanism through which CD4 coordinates everything downstream. Interleukin-2 drives T cell proliferation, expanding both CD4 and CD8 populations rapidly. Interferon gamma activates macrophages, significantly enhancing their phagocytic and antimicrobial capacity. Interleukin-4 and interleukin-5 support B cell differentiation, proliferation, and antibody class switching, directing which immunoglobulin type is produced. CD4 cells also provide direct co-stimulatory signals to CD8 T cells. This licensing is critical. Without CD4 help, CD8 cells can mount a primary response, but they cannot form durable immunological memory. the secondary response is severely impaired. This dependency is clinically significant. When CD4 function is compromised, as in HIV infection, the downstream consequences cascade through every arm of adaptive immunity. Antibody responses weaken. Macrophage activation falls. CD8 memory formation fails. The entire adaptive system loses its coordinator. CD8. T cells are cytotoxic T lymphocytes. Their function is the direct, targeted elimination of cells that have been compromised, whether by viral infection, intracellular bacterial infection, or malignant transformation. The mechanism begins with surveillance. Every nucleated cell continuously processes its internal proteins through the proteasome, breaking them into peptide fragments. These fragments are loaded onto MHC class I molecules and transported to the cell surface. In a healthy cell, these displayed peptides are derived from normal cellular proteins. CD8 T cells passing through recognize this as self. No response is triggered. In a virally infected cell, viral proteins are being synthesized in the cytoplasm. Viral peptides enter the MHC class I pathway and appear at the cell surface. A CD8 T cell with a matching receptor recognizes this altered display. Recognition initiates the cytotoxic sequence. The CD8 cell forms a tight junction with a target cell, called the immunological synapse, and releases the contents of its cytotoxic granules in a directed manner. Perforin is released first. This protein polymerizes in the target cell membrane, forming transmembrane pores. Through these pores, granzymes enter, serine proteases that directly activate caspase cascades inside the target cell. Caspase activation triggers apoptosis, the programmed, orderly self-destruction of the infected cell. The cell dismantles itself from within. The virus, trapped inside, is destroyed with it. Critically, adjacent cells displaying normal MHC class I peptides are completely unaffected. The CD8 cell then disengages and moves to the next target. This is the defining characteristic of CD8 cytotoxicity: surgical precision at the cellular level. Following pathogen clearance, the expanded effector T cell population undergoes a contraction phase. The majority of effector CD4 and CD8 cells, no longer needed in large numbers, undergo apoptosis. A small fraction, approximately 5 to 10%, survive as long-lived memory T cells. Memory cells are phenotypically and functionally distinct from naive T cells. They have lower activation thresholds, respond more rapidly, and produce effector molecules faster on re-exposure to antigen. A naive T cell encountering a pathogen for the first time requires 5 to 7 days to mount a full response. A memory T cell responds within hours. CD4 memory supports faster, stronger coordination of secondary responses. CD8 memory delivers immediate cytotoxic activity on antigen re-exposure. This accelerated secondary response is the immunological basis of vaccination, and the reason some infections confer lasting natural immunity. In healthy individuals, the CD4 to CD8 ratio in peripheral blood is maintained at approximately 2 to 1. This ratio is clinically measurable, and its disruption is a marker of immune dysfunction. In HIV infection, the virus selectively targets and depletes CD4 T cells. As counts fall below 200 cells per microliter, the clinical threshold for AIDS, adaptive immune coordination collapses. Infections that a competent immune system eliminates routinely become fatal. In cancer, tumor cells frequently upregulate PD-L1, a ligand that binds the PD-1 inhibitory receptor on CD8 T cells. This interaction delivers a suppressive signal, driving CD8 cells into a state of functional exhaustion within the tumor microenvironment. In autoimmune disease, dysregulated CD4 subsets, particularly Th17 cells, drive inflammatory responses against self-tissue. Each of these failure modes has a distinct mechanism. Each represents a separate case, and each is the subject of the next videos in this series. CD4 and CD8 T cells represent the two primary effector arms of adaptive immunity. CD4 coordinates, reading antigen, releasing cytokines, directing every downstream response. CD8 executes, identifying compromised cells and eliminating them with molecular precision. Neither functions fully without the other. All sources are linked in the description: Nature Immunology, Signal Transduction and Targeted Therapy, Nature Reviews Immunology. Every claim is referenced and traceable. One question. The CD4 to CD8 ratio is already measured in HIV monitoring. Should it become part of routine blood work for everyone, as a standard immune health marker? Tell us in the comments. Next, we examine HIV, specifically how a virus evolved to target the one cell that coordinates everything. Case 0. Every mystery begins somewhere.